You are breathing it, drinking it, eating it, and it is already inside your brain. In February 2025, a study published in the journal Nature Medicine by researchers at the University of New Mexico found something that should have stopped the world. Human brains collected at autopsy contained microplastics at concentrations 7 to 30 times higher than any other organ in the body. The concentration had increased by 50% compared to brain samples from just 8 years earlier. Brains of individuals diagnosed with dementia contained up to 10 times more plastic fragments than healthy brains. This is not a projection. This is not a model. These are measurements from human tissue. And that is only one of the 3 contamination systems we are investigating today. This is the Brain Care Podcast, Season 2. I am the doctor. Fluoride is added to public drinking water in multiple countries, including the United States, with the stated goal of reducing dental cavities. The World Health Organization guideline limit is 1.5 milligrams of fluoride per liter of drinking water. The U.S. enforceable limit is 4 milligrams per liter. That is more than double the WHO threshold. In 2025, a meta-analysis published in JAMA Pediatrics synthesized 74 epidemiological studies involving 20,132 children. The finding was consistent across analyses. Higher fluoride exposure was associated with significantly lower IQ scores in children. The pooled standardized mean difference was 0.45, which translates to approximately 7 IQ points on a standard scale. This result held even in the subset of studies rated as low risk of bias. The 2024 National Toxicology Program Monograph, a U.S. government review, concluded with moderate confidence that higher fluoride exposure is associated with lower IQ in children. The pineal gland is a small endocrine structure located at the geometric center of the brain. It produces melatonin, the hormone that regulates your circadian rhythm, your sleep architecture, your immune synchronization, and your antioxidant defense. A 1997 doctoral study at the University of Surrey by researcher Jennifer Luke found that the pineal gland has a higher concentration of fluoride than any other soft tissue in the body, and that this accumulation is directly correlated with calcification. Pineal gland fluoride levels in aged cadavers were measured at 296 milligrams per kilogram. Muscle tissue of the same subjects measured 0.5 milligrams per kilogram. The same study found that high fluoride animal subjects excreted significantly less melatonin than low fluoride subjects, with the most measurable suppression occurring during the developmental window before sexual maturation. A 2025 review published in an international research journal confirmed the association between pineal gland calcification, fluoride exposure, and declining melatonin production, and linked calcification to increased oxidative stress and mental health disruption, including anxiety and depression. Over 60% of adults show measurable pineal gland calcification on brain imaging. Most of them have never been told what it means for their brain biology or what put it there. Microplastics and nanoplastics are plastic particles smaller than 5 millimeters. Nanoplastics are smaller than 1 micrometer, invisible to the naked eye. They enter the body through ingestion, inhalation, and dermal contact. They have been detected in human blood, breast milk, placental tissue, and fetal tissue. And as the 2025 Nature Medicine study confirmed, they are accumulating in human brain tissue at concentrations that are increasing year over year. A 2025 systematic review published in PMC synthesizing 234 original research articles found that microplastic and nanoplastic exposure is associated with neuroinflammation, microglial activation, dendritic spine loss, altered mitochondrial function, and compromised blood-brain barrier integrity. The mechanism at the barrier level involves the suppression of tight junction proteins, specifically zonula occludins 1 and occludin, which are responsible for keeping the blood-brain barrier sealed. When these proteins are disrupted, the barrier opens. A 2025 review in Frontiers in Toxicology confirmed that nanoplastics can cross both the blood-brain barrier and the placenta, gaining access to the central nervous system and the developing fetal brain. Once inside, they trigger multiple simultaneous damage pathways: oxidative stress, persistent neuroinflammation, mitochondrial dysfunction, disruption of neurotransmitter systems, and direct neuronal damage. Critically, nanoplastics have been shown to promote the aggregation of alpha-synuclein, the protein whose abnormal accumulation is directly implicated in Parkinson's disease. Animal studies have demonstrated that microplastic exposure causes dose-dependent spatial memory deficits, hippocampal oxidative damage, disruption of the BDNF and CREB neuroplasticity pathways, and reduced acetylcholine levels. These are not isolated laboratory findings. Vitamin E intervention in animal models reversed both the biochemical damage and the learning deficits, establishing a clear mechanistic link between microplastic exposure and cognitive dysfunction. The most alarming variable is not the toxicity at high doses, it is the trajectory. 50% more plastic in human brains in 8 years, and no meaningful regulatory response. Lead, mercury, arsenic, and cadmium are classified as priority neurotoxins by every major global health authority. There is no safe threshold. There is no biological function that requires them. A 2022 study published in PMC analyzing NHANES population data found that cadmium and lead concentrations were significantly associated with decrements in performance. Across all 3 cognitive test categories administered, the associations held after adjustment for confounders. A 2021 PMC review on lead toxicity found that 37% of children living in high-risk lead pollution areas showed measurable cognitive dysfunction. Lead disrupts hippocampal NMDA receptor subunit expression, altering synaptic plasticity and spatial learning. In ways that persist into adulthood, lead disrupts hippocampal neurogenesis. The damage starts in childhood. Lead exposure during early childhood alters the pattern of newly born hippocampal cell differentiation. This produces long-lasting impairments in spatial learning and executive function. The damage does not resolve when the exposure ends. It is encoded into the developing brain architecture. Mercury, specifically methylmercury from contaminated fish sources, has been shown in research published in 2026 in the journal Neurologia I Neurochirurgia Polska to trigger the same cascade of molecular events observed in neurodegenerative disease: release of amyloid beta, enhanced tau phosphorylation at sites identical to those observed in Alzheimer's disease and frontotemporal dementia, calcium homeostasis disruption, mitochondrial dysfunction, and oxidative stress sufficient to kill cortical neurons. Mercury shows a pronounced affinity for thiol and selenol groups in the nervous system. These are the molecular anchors of your antioxidant defense. When mercury occupies them, your neurons lose their primary protection. Arsenic is present in groundwater in numerous regions globally. A 2025 review published in PMC analyzed 24 studies on arsenic exposure and cognitive performance in children, finding a consistent inverse relationship: higher arsenic exposure, lower cognitive performance. A comprehensive review published in Science Direct in 2025 confirmed that arsenic disrupts astrocyte signaling pathways, impairs brain homeostasis, and contributes to cognitive loss and neurotoxicity through both direct neuronal damage and systemic inflammatory mechanisms. These are not emergency poisonings. They are low-dose, long-term, cumulative processes, and their endpoint is a brain that functions below its designed capacity, while the individual believes that is simply how they are. File number 003, subject Clair Patterson. In 1956, Clair Patterson, a geochemist at the California Institute of Technology, was trying to calculate the age of the Earth using LED isotope ratios in ancient meteorites. To do this precisely, he needed to measure natural background levels of lead in the environment. What he found instead permanently changed the history of public health. The lead levels in modern human blood, bones, and tissue were 100 times higher than in ancient Peruvian remains predating industrial civilization. Patterson realized immediately what this meant. The global environment had been contaminated with industrial lead at a scale that was altering the baseline lead concentration in every living human being. The primary source was tetraethyl lead, a gasoline additive introduced by General Motors and Standard Oil in 1923. Patterson published his findings in the journal Science in 1965. He called for a complete ban on all uses of LED in consumer products and gasoline. What followed was a 20-year coordinated campaign by the LED industry to discredit his research, suppress his funding, and prevent his findings from influencing policy. The Ethyl Corporation and the LED Industries Association contracted scientists to counter Patterson's data. They pressured the U.S. Public Health Service. They lobbied regulatory agencies. They called Patterson's work alarmist. They produced industry-funded studies that found no harm at levels they themselves determined to be acceptable. Patterson was removed from a National Research Council panel on atmospheric lead contamination after objecting to industry representation on the panel. He lost industrial funding. His laboratory was defunded. He was professionally isolated for years. Leaded gasoline was not fully phased out in the United States until 1996, 40 years after Patterson first published the proof. The accumulated lead exposure during those 40 years has been linked in subsequent epidemiological research to measurable population-level IQ reduction. elevated crime rates correlated with childhood lead exposure and generational cognitive impairment in communities near industrial lead sources. The science was available, the decision was made to ignore it. Patterson died in 1995, 1 year before the ban he had advocated for in 1965 finally took effect. He never received a Nobel Prize. His story is not a historical footnote. It is the operating manual for how neurotoxic substances are normalized. Identify the contamination, fund counter-science, delay regulation, let the exposure become baseline, call anyone who questions it alarmist. The same peer-reviewed science that confirms this contamination also maps the countermeasures. Let me be precise. Pay close attention to the following evidence-based protocols. The primary intervention is source reduction. Filtered water removes fluoride and heavy metal contamination that tap water does not. A high-quality reverse osmosis or activated alumina filter reduces fluoride concentration measurably at point of use. Reducing ultra-processed foods, single-use plastics, plastic packaging, and plastic-lined beverage containers reduces ongoing microplastic and nanoplastic ingestion. This is not optional self-care. This is primary prevention. The body has endogenous heavy metal detoxification pathways. The most important involves glutathione, your primary intracellular antioxidant, which binds and facilitates the excretion of heavy metals through biliary and renal pathways. A 2019 review published in PMC confirmed that chelation therapy using ETA was able to remove all measured toxic metals from neurodegenerative disease patients with no adverse effects. The same review documented improved neurological symptoms following removal of aluminum, lead, and mercury burdens in multiple patient populations. For nutritional support of natural chelation pathways, a 2019 study published in PMC found that chlorella vulgaris extract combined with nutraceutical aminosulfurate supplementation produced measurable reduction in heavy metal levels over 90 days. Chlorella reduces cadmium and dioxin levels through biosorption and induction of metallothionein-like proteins. Note, cilantro-based claims for chelation remain unverified in human clinical trials and should be treated with caution. The evidence for chlorella-based protocols is more robust. For microplastic neurotoxicity specifically, the evidence points to antioxidant supplementation. Vitamin E was shown in animal studies to reverse both the biochemical damage and the cognitive deficits associated with polystyrene microplastic exposure by addressing the oxidative stress at the cellular level. Curcumin has been identified in recent research as reducing oxidative stress from micro and nanoplastic exposure. Probiotic supplementation has been shown to suppress plastics-induced neuroinflammation. These are not speculative wellness recommendations. These are the interventions being studied in active research programs. For the pineal gland specifically, reducing fluoride exposure through filtered water and supporting melatonin production through consistent sleep hygiene, darkness exposure protocols, and antioxidant support represents the most evidence-aligned approach available in 2026. Your genome did not evolve in a plastic-saturated, fluoridated, heavy metal-contaminated environment. But your epigenome is adaptive. The toxins entered gradually. The recovery also proceeds gradually, and it begins with knowing what you are recovering from. You were not told about the microplastics in your brain tissue. You were not told about the pineal gland calcification or what it does to your melatonin and your sleep. You were not told about the decades-long campaign to normalize the contamination of your nervous system. Because the systems that benefit from your continued exposure have no interest in your biological sovereignty. Understanding your specific constitutional vulnerability to environmental toxins is the first step toward targeted intervention. The neuroenergetic blueprint of the 5 elements identifies your elemental constitution, your dominant detoxification profile, and your personalized exposure risk pattern. It gives you a starting protocol that accounts for your biology specifically, not a generic list of supplements that ignores who you are. €29. The link is in the description. For those ready for a clinical-grade environmental detox protocol, the Elemental Performance Lab includes heavy metal burden assessment, glutathione pathway optimization, microplastic mitigation strategies, and 12 weeks of physician-led supervision. The link is in the description. Next episode, Pharmaceuticals and the Business of Sickness, the chemical foundations of modern medicine, who built them, and what was decided about your brain before you were born. This is the Brain Care Podcast. Season 2, I Am the Doctor. Protect your brain, because no one else will.